I used to wonder how my cells grow. They divide their nucleus. Wonder how my cells grow. Mitosis the nucleus. Mitosis is dividing. So each cell has same old thing. Meiosis is dividing. So you cells can make offspring. Ever since they left G2.
Our bodies are made out of thousands of living particles called cells. But what happens when one or more of these cells die? How does our body regenerate these cells in order to keep us from dying too? That's where cell division comes in. Cell division is divided up into 5 main stages: mitosis, which consists of prophase, metaphase, anaphase, and telophase; cytokinesis, gap 1, synthesis, and gap 2. Interphase is the longest phase of the cell cycle, and the cell remains in this phase for about 78% of its lifetime. This phase consists of gap 1, synthesis, and gap 2.
Shown here is a simplified drawing of the cell going through the gap 1 phase. The outer ring is the plasma membrane, the inner ring is the nuclear envelope or nuclear membrane, and inside the nuclear envelope is a dispersed form of DNA known as chromatin. Notice that the DNA is not in the shape of a chromosome the way it usually is depicted. This is because the DNA is yet to take this form. Depicted in purple is a centrosome of the cell containing 2 centrioles. During the G1 stage, a cell grows as it normally would, whether or not it is undergoing cell division. The cell grows in size, proteins are synthesized, and organelles, such as the mitochondria, replicate.
As a cell moves from the gap 1 stage to the synthesis stage, it must check to make sure it meets the requirements for cell division. Essentially, the cell must decide whether or not to divide. The cell checks for numerous things. The cell must check its size to ensure that it is big enough to divide. It also must check if it has enough nutrients and energy reserves to support 2 cells after it divides. It also checks whether it is receiving any signals from its neighbors or the body. For example, if there is already a sufficient amount of cells in the body, the surrounding cells might chemically signal a cell not to divide. Additionally, it checks its DNA before it enters the synthesis phase. If there is any damage to the DNA, the cell should not divide to prevent this damaged DNA from spreading. If any of these requirements is not met, the cell will move to the G0 phase, where it may either stay resting or continue to divide when conditions improve.
Next is the synthesis phase. In the synthesis phase, the DNA inside the nucleus of the cell replicates, and each DNA strand is joined together to its duplicate at the region known as a centromer. Each strand is now known as a sister chromatid, and each pair of the sister chromatids is known as a chromosome. However, the DNA is still not condensed into the way a chromosome usually looks. The next thing that happens in synthesis is the replication of the centrosome. Before synthesis, the cell has 1 centrosome, and after synthesis, the cell has 2 centrosomes, each containing 2 centrioles.
In the gap 2 phase, there's more cell growth, and the cell synthesizes more proteins that might be needed for cell division, such as microtubules. Before the cell enters the mitosis phase, it must pass through another checkpoint. This time, the cell makes sure that no DNA has been damaged during the process of DNA replication, and that all the DNA has been copied completely. If the cell finds any repairable errors in the DNA replication or any repairable damage to the DNA, it will stop immediately and fix these errors before continuing the cell cycle. However, if it were to find any unrepairable damage to the DNA or unrepairable errors in the replication, the cell will undergo apoptosis or programmed cell death in order to prevent harming the organism.
Mitosis is a very short process compared to interphase, and the cell only spends about 20% of its life in this phase. Mitosis is essentially the process in which the cell divides its nucleus. It consists of 4 subphases: prophase, metaphase, anaphase, and telophase.
The first subphase of mitosis is prophase. In prophase, the chromosomes condense and become shorter and thicker. Even though they are condensed, they are still called chromosomes. This is the X shape that is usually used to depict chromosomes. Next, the nuclear envelope breaks down. The centrosomes, which were replicated during synthesis, also migrate to opposite sides of the cell.
After prophase comes metaphase. In metaphase, the chromosomes line up along the middle of the cell and long protein fibers called microtubules form spindles, which extend from the centrosomes to the chromosomes. As metaphase comes to a close, a cell must pass a third checkpoint. At this third checkpoint, which is also known as a spindle checkpoint, the cells make sure all the chromosomes are attached to the spindle fiber on both sides at the metaphase plate. Because the process of anaphase is highly irreversible, the cell must pass this checkpoint in order to move on.
The function of these spindle fibers is seen in this next phase, called anaphase. As soon as the spindle fibers attach to the sister chromatids at a region called the kinetochore, they begin to pull sister chromatids to opposite sides of the cell, as well as push the centrosomes away from each other. After they are separated, the former sister chromatids are each called chromosomes. Because the spindle fibers from each centrosome are pushing on each other, the cell also begins to elongate. The division of genetic material during anaphase is called karyokinesis.
In telophase, the last formal stage of mitosis, the spindle fibers detach from the chromosomes, and the chromosomes on both sides of the cell begin to unwind back into a chromatin form. 2 nuclear membranes form
After mitosis, a process called cytokinesis takes place. It is the last stage in the cell cycle and consists of the dividing of the cytoplasm. Cytokinesis is a process which occurs differently in animal and plant cells. First, we'll take a look at how it happens in animal cells. First, a cleavage furrow forms. A cleavage furrow is an indentation of the cell surface. Then, a ring of microtubules forms near the equator of the cell. As this ring shrinks, the space connecting the cytoplasm of both sides of the cell gets smaller and smaller until eventually the cytoplasm splits into 2, and the initial cell has become 2 different cells. In plant cells, the process is slightly different. In plant cells, no cleavage furrows form due to the sturdy cell wall keeping the cell in its shape. Membrane-bound vesicles line up at the equator of the cell. Slowly, these vesicles merge together, creating what is known as a cell plate in the middle of the cell. This cell plate fuses with the cell wall, finally creating two separate cells. Cancer is a serious condition which is signified by uncontrolled cell division and growth. It can be caused for a variety of factors, such as oncogenes. Oncogenes are genes which are usually switched in the off position, but when altered or influenced can be switched on and can seriously harm the body. Cancer can also be caused by various carcinogens, such as radiation, viruses, and certain chemicals. These carcinogens attack the DNA of a cell and disrupt control of cellular division and mitosis. Cancer can be caused when cells cannot respond to certain growth regulators in the body. One example of a growth regulator would be the inability to maintain homeostasis. As a cell's volume increases, it needs to take in more nutrients as well as produces more wastes. However, the only way these wastes can leave the cell and these nutrients can get in is through the plasma membrane. Assuming that the cell is a sphere, as shown in these two equations, the volume of the cell will grow at a larger rate than the surface area of the cell. This means that the cell's produced wastes and required nutrients will increase at a larger rate than the amount of space it has to excrete these wastes and take in these nutrients. This will result in the cell having a difficult time maintaining homeostasis. This difficulty maintaining homeostasis will trigger cell division.
Shalone is a glycoprotein which inhibits mitosis in the specific tissue which secretes it. If it is absent, cell division will be triggered. If present, it inhibits cell division. Hormones are another type of chemical signal.
Cell growth can also be inhibited when cells grow too large and begin to touch each other. This lets cells know that there's no space for new cells and that they should not divide.
Cancer cells may not be able to respond to these signals and therefore begin to grow uncontrollably.
In regards to the checkpoints described earlier, cancer cells will tend to ignore these checkpoints and continue to divide even if they don't meet the requirements at that checkpoint. The specific type of uncontrolled cell growth associated with cancer is called a malignant tumor.